we did talk about how those different structure of the brains are formed, right? Now we want to go to the spinal cord, okay? And we know that spinal cord will be induced by what? What will be beneath spinal cord? Coda mesoderm, head mesoderm, or precaudal plate? Coda mesoderm is not a cord. Not a cord. Not a cord. Right. So we want to talk about how the spinal cord is specified, okay? And we are going to talk about specifically the dorsoventral axis of neural tube. Remember, this is still neural tube. This has not formed spinal cord yet, right? So what is happening there? If we look at neural tube. So this is the neural tube right here, and this is the central cavity right there. And this is the dorsal side and the ventral side. Now, in spinal cord, so what is happening? Let me draw it a little bit here. So this is your spinal cord. So all your sensory signal, for example, someone touch your hand, right? And that signal goes there. That signal is going from your body to this group of neurons. These groups of neurons are called dorsal root ganglion. These are sensory neurons. So what happened, these sensory neurons from here, they need to send signal to here to your spinal cord. This is your spinal cord. So now look here. So this is, we are talking about how this connectivity is made. And what are the neurons in the neural tube which are ultimately going to give you the spinal cord. So what is going to happen right here at this place, you will have the sensory neuron entry. So from this DRG, the neuron are going to send a projection right here. Okay? And then right here, you will have the group of neurons, what we call spinal neuron. Now, think about if you put your hand in the fire. So what is the immediate reaction? You don't want to keep it there, immediately move it out. So it means that signal has gone from here to your spinal cord. Your spinal cord has processed that and sent the information to the muscles here to put it out, right? So what is happening there, these spinal neurons are getting that, and right here on this side, right here are the neurons, this group of neurons, these are called motor neurons. So motor neurons are controlling your skeletal muscle, they are on your will, right? So now, this signal come here, from this signal will be sent to these neurons, other color, these neuron will send it to other neuron ultimately here. So these neurons which are here, these are called interneurons, and these neurons are actually talking to the spinal. They essentially are doing the relaying the information. They relay the information. And then again you will have certain interneurons here as well. So this is actually how the spinal cord, if you cut a cross section, will look like. Now the question here is that this is in the adult when you are developed, embryo is formed. So how this structure is formed, how these neurons are specified that, hey, you have to be the spinal neuron, you have to be the interneuron, you have to be the motor neuron. So motor neurons are probably critical for our survival. Think about, I just give you the example of, putting hand in the fire, but you're walking, you're breathing, everything is controlled by motor neuron. You have heard of this disease called ALS, right? So ALS is the problem of the motor neuron. So how these neurons are formed, that's what we want to talk about. And that is what we are going to say is the dorsal ventral axis of neural tube, and we're talking about the formation of the axis. So this dorsal ventral axis is specified by the signals which are coming from the surroundings. Okay. So remember that this was our neural tube. So right here is the neural tube, right? Which we talk about how it closes, right? And what we have here on this side. What is this? Epidermis. And then, what is this? Notochord. So the two signaling centers which are going to tell that which kind of neurons are going to form here, those are epidermis and notochord. So notochord, what does notochord secrete?
Noggin and sonic hedgehog.
So noggin and sonic hedgehog, right? So right here, in this case, in the formation of this axis, the sonic hedgehog is playing a critical role. So notochord is going to secrete sonic hedgehog, writing here SHH, okay? And the epidermis, what does epidermis secrete? BMPs, right? So epidermis is going to secrete BMP7 and BMP4, right? Now these proteins, BMP7 and BMP4, these are actually belong to a family of transforming growth factor beta, what we call TGF beta. So these are from that family. So what is going to happen? Now, epidermis is secreting here on these cells lot of BMP4 and BMP7 is being secreted here. Okay. Now these cells here where it's secreted, which are getting it, these are called roof plate cells. So this form a roof plate. Okay. Now the cells here, what is the name of these cells? Mediolateral hinge, right? So these are the MLH cell, mediolateral hinge cell. So these cells are getting a lot of sonic hedgehog. So a lot of BMP4, BMP7 coming here, and a lot of sonic hedgehog coming in these cells. So what it does actually, when you have a lot of BMP4, 7 coming here, a lot of sonic hedgehog coming here, these cells of roof plate and mediolateral hinge cell, now we also call them floor plate cells, because those are the floor plate. So these cells of roof plate cell and the floor plate cell, they actually start acting as secondary signaling center. Okay, so because of this, a lot of sonic hedgehog coming here. So what happened that these cells right here and these cells here, they become secondary signaling centers. So these two are now new secondary signaling center. So you have these, your roof plate cell I'm just writing RPC and your floor plate cells. They become secondary signaling center. And what they do, they actually start secreting the roof plate cells. They start secreting more amounts of TGF-beta proteins. So there is okay, so what happened? Right here, the cells which are here on this area, these cells start secreting a lot of BMP 4, 5, 7, dorsalen, and activin, cell which are right here. And then there are the cells next to it, cell in this area, which is all blue here. These cells, they start secreting BMP5, dorsalen, and activin. And the cell next to them, which are right here, these cells secrete dorsalen and activin. So what is happening? So what we are saying that we have this epidermal cell, they started secreting BMP4 and 7 on these roof plate cells. Because of that, these cells forming the secondary signaling center. And we are saying then these cells, they secrete variety of TGF-beta family protein. On the top, you have BMP4, 7, dorsalen, and activin. Then you have BMP5, dorsalen, activin, and then later you have dorsalen, activin. Now, these are secretory factors. So what happened, that these factors will be released like this from here to here. So they will be secreted. And then there will be gradient, like more of these here, as you going here, less, more of these here, because they are secreted here, more amount here, slowly going down. Making sense? Now, what is happening on this side? On the floor plate cell, another center was created because notochord secreted a lot of sonic hedgehog. And these floor plate cells, they actually start secreting sonic hedgehog. So right here, they are secreting a lot of sonic hedgehog. And from here, the sonic hedgehog will be secreted. It will be a gradient here on this side also. Like this. Making sense so far? So we are saying what is happening? Because of the epidermal BMP4 and 7, a new signaling center was created at the roof plate. Because of notochord secreted sonic hedgehog, a new signaling center was secreted in the floor plate. The floor plate secretes sonic hedgehog, and more sonic hedgehog near the ventral, and then you have a gradient. Whereas on the dorsal side, you have all the transforming growth factor proteins are being secreted, and what they are doing, they are making a gradient here. You have more TGF family here, coming here, less, more, less. Making sense so far? Now what happens? Now what these factors are doing, they are now going to make specific gene expression patterns of transcription factors in these areas. Okay? So because of that, what is happening, that lead to a situation a little bit bigger I'm drawing. So the area where you had the huge amount of transforming growth factor, that area right here is going to start expressing the transcription factor PAX7. So PAX7 protein is made here a lot. So when I'm saying expression, I'm not talking about gene expression, I'm talking about protein expression. So I have more and more protein there, PAX7 there, right? And then after that, in these areas, as we are going from here to here, we will have a gradient of PAX6. So we'll have PAX6 here. So I'm talking about protein again, remember that. So you will have more and more PAX. Again, a gradient will be there, like this. So more PAX6 here, less PAX6 here, all depending upon this gradient which was coming from transforming growth factor. Now, on this side, where we had a lot of sonic hedgehog on this side, you will have another expression of the gene. Again, you will have more on this side, and again, it will slowly, more here, and slowly going down like this. All these areas will have another protein called NKX6.1. Now, this expression pattern of these transcription factor proteins is going to induce specific proteins in the cells here, and that is going to give specificity to the neurons. So what is going to happen? Here you will have, because this is the dorsal part, right? This is the ventral part. So you need to have those neurons, and you will have D1, D2, V0, V1, V2. Now, here is interesting thing. If you look here, this is the area where, in this area, you can see that there is an overlap of PAX6 and NKCX.1. Where both PAX6 and NKX6.1 are made, those neurons right here, these neurons where you have both of PAX6 and NKCX1, they are going to become motor neurons. Okay, and the neuron here which have only NKX6.1, they will become V3 neurons. So remember, in the beginning I told you that we have a polarity here. We said right here the sensory neurons are going to come, then you will have all the interneurons there, then you will have the motor neuron, right? So this polarity is established because of the signaling which is coming from epidermis and notochord. So what we are saying that the specification of the neural tube is established by the two signaling area, epidermis secreting BMP4 and BMP7, notochord secreting sonic hedgehog. This secretion of BMP4/7 create a new signaling center on the roof plate. Signaling from the sonic hedgehog through notochord make a new signaling center in the floor plate. Now, the roof plate cells, they start secreting huge amount of transforming growth factor beta family proteins. On the top, you have all BMP4, 5, 7 dorsal activin. Here, slightly less, you have BMP5, dorsal activin, and then you have dorsal activin. Now, these transforming factor or secretory factor, they move down and basically a gradient is formed. Higher transforming growth factor on the dorsal side, less on the ventral side. Another signaling center which is created here, that is secreting sonic hedgehog, very high sonic hedgehog, and it tapers down as it goes toward the dorsal side. And we are saying that this interaction of transforming growth factors, beta family, and the sonic hedgehog family, they'll start expressing different kind of transcription factors there, and those transcription factor protein are going to activate the genes which are specific for specific types of neurons. And we are saying on the top you will have PAX9, then PAX6, and there will be a gradient of PAX6, high PAX6, low, 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 going down. Same way we will have ventral side, NKX6 start expressing, again there will be a gradient, high on ventral side, less on the dorsal side. So at the point where the cells are showing both PAX6 and NKX6.1, those will become the motor neuron. And depending upon the concentration, very high PAX6, you want D1 interneuron, less PAX6, D2 interneuron, lesser than that, V0, lesser than that, V1, then V2. And very high sonic hedgehog, V3. Now, how do we know that this, what we are talking, is real thing or not, right? So if you do an experiment, you take a notochord, developing neural tube, and you have this notochord here, right? You take another mouse embryo, and you take notochord from that mouse embryo and put it here. So what is going to happen, you will have another motor neuron column established here on this side. So that means the notochord signaling is actually essential for the formation of the motor neuron. Making sense or not? Okay, talk to each other for a minute, then we'll go for 2 minutes, then we'll go further. So talk, and then we'll go further.
Receive and send signals to the spinal cord, and to do that the signal goes into the sensory neuron entry at the spinal neuron, and that travels through interneural
So you will have actually two centers here, right? So initially you should have motor neuron here, right? But they put a new motor column here. So you have one motor column here, one motor column here, one here.
And they're specified by their surrounding signals. The
So that
epidermis
means the
secretes
sonic
BMP7
hedgehog
and
signal
4,
is
which are transforming growth factor beta.
Or you can do the same thing with the epidermis.
And
You can
those
bring
form the
something with
root
the
plate.
PMP secreting here, so you can change
And
that
then the
also.
notochord, which secretes sonic
Right.
hedgehog. At the medial lateral hinge is also the floor plate.
Any doubt here? No?
And those form secondary signaling.
So if you had, instead of notochord, if you put epidermis here, you're going to change that also, right? Any doubt here? Okay, good. Okay, so let me show you this thing. This is from the. So I told you that that the neurons are sending projection here, right? So how does the neuron know that I have to send projection here? And how does your sensory neuron know that, hey, this projection should go to DRG, and from DRG it should go to the spinal cord, right? Because the cell body is residing there in these columns, but all the wires are here. So how does that happen? Let me show you this thing so that you can appreciate it. So this is done. This was a long time ago. I had a student, Danny Varga, in my lab. So he took it. So this is 24-hour time lapse picture showing you how actually the axon grow, right? So look here very carefully. Just turn this off for a second, so you can actually look and appreciate it, what is happening there. Try to make it bigger. No. Okay, will not go bigger than that, that's okay. Look here. Hopefully it will work. Pay attention here. Same way here. So did you see what happened? These are axons, right? On the tip of axon, something was doing like this, and going further, right? So that area on the tip of the axon, if this is the axon, and this area, which was kind of like this was kind of like going like this, kind of like this. That area is called growth cone. That is called growth cone. And this growth cone is critical. Now, what we are talking about, we're talking about how does the neuronal axon know where to go? That is the question we want to answer. Where to go? That is, what is the target? First, what is the target? And the second question is, how to reach target? How to reach target? And then, even I reach at the target, how to select the address, and what is address, I'll explain you. Now think about when we are developing. So we did talk about the development of the nervous system, right? But having neuron in the spinal cord or in the brain has no meaning. Those should be able to do what they want to do. And if they want to do, they should be, if we know they control muscle, they control everything, they should be able to reach the target. And how that target is selected. So target selection, what is the target? So think about why we go to work, or why you come here to study. You come here because you want to get the education, right? Same way, we come here because of course we love to teach, but that teaching is also giving us money, right? So we go for something. So same way, when a neuron decide, for example, this neuron, it decide, hey, I want to come here to the skin. There should be in the skin something that is good for neuron. And usually, the targets, they secrete trophic factors. Trophic factors, in a simple word, are called survival factors. So they go for that. So each neuron is looking for a specific survival factor that is secreted by a specific type of the cell. But then the thing is how to reach there. And that's what we are talking about, the pathway selection. And we'll talk mostly today on this pathway selection. That which route to take. For example, think about that if you're going from here to Liberty Square, okay? So you will go out from here, you will take the main road of the college that is going out, but then you should know you want to move right or left. You cannot move left till you get the signal turn left, right? Or if there is red light, you stop there. But you cannot just, take the car and go down the hill. You have to follow a road. The same way, the neurons, when they're moving, they are moving on the roads and they are following those stop, go, turn left, turn right signals. And what are those signals? Those signals are, so pathway selection, we're talking about the signals for that pathway selection. What are those signals? those signals can be attractants, or those signals can be repellents, which are attracting them and repelling them. And there are molecules which are secreted by the surroundings. Now let me explain what are those. So you can have proteins what we call ephrins. So when the neurons are moving, surrounding cell, axons are moving, surrounding cells are producing these chemicals. So ephrins are chemorepellent. That means they are going to tell neuron, axon, don't come here. But how they can tell? They can tell only if the growth hormone has the receptor for that chemorepellent. So the receptor for these chemorepellent is TPH. And we call them ephrin proteins actually. That's why we wrote them. Ephrin proteins. Now there can be many type of ephrin. I'm just writing a big class so that you just know that. And then there are other class of protein, what we call semaphorins. This is another class of protein. These are also chemorepellents. These are also chemorepellents. And their receptors are collapsins. No. They are neuropillins. Their receptors are neuropillins. Then you have natrins. These are attractant. And their receptors are DCC and DSCAMs. Then there is fourth category. And the fourth category here is SLIT. And the receptors are ROBO, R-O-B-O. And these are also repellent. So, like the road, when you go, you have go signal, you have stop signal. And those go and stop signals are secreted by the cells surrounding areas. And those signals, these chemicals which are secreted by the surrounding cells should be received by the growth hormone. So this is our growth hormone. So growth hormone should have receptors to answer those signals. And we are saying there are four classes or four families of proteins are there which can act as either chemorepellent or chemoattractant. We are saying ephrins, semaphorin, and SLIT. These two classes are chemorepellent. They're telling the neurons or the axon, don't come here. Whereas natrins, they are telling, come here. So this is kind of a green light, all other are the red light. So to get that, understand that light, you should have those receptors. And we are saying ephrins have EPH or F receptors. F receptors should be on the growth hormone. Semaphorin should have neuropillin receptors on the growth hormone to understand that signal. The natrin, that signal can be understood if you have DCC and DSCAM receptor. And SLIT should have a receptor called ROBO. So receptors are on the growth hormone, and these proteins are secreted by the surrounding cells. That is making sense so far? Now let me give you two examples how actually this signaling work, so that we can understand it better. Okay, so now we are talking about how they go to target, right? And we are talking about the targets of motor neurons, how those targets are made. Now, once a cell becomes motor neuron, so motor neuron cell has become, that motor neuron say, okay, where is my target? Is it the thigh? Is it the forearm? Or it is the tongue? How that target is decided? That target decision is made while neurons are developing. So, think about that this is a motor neuron form, this is another motor neuron, this is another motor neuron. These are the motor neurons. And these motor neurons are made in the neural tube. We just explained how they are made, right? Then you have another motor neuron form, another motor neuron, another motor neuron. I'm just trying these 6 neurons. Now, each of these motor neuron will have a specific cell surface protein and so these cell surface protein, these are neuronal cadherins. Now, here if you pay attention, these 3 cells, 1, 2, 3, they are expressing similar type of neural cadherin, right? These 3 are expressing similar type, these 3 are expressing similar type. So the neuron expressing similar type of cadherin, they bundle together, okay? So we will have a group of neurons right here, 1, 2, 3, 1, 2, 3, 1, 2, 3. And if they have similar cadherin, for example here, they will bundle together, they are going to bundle together, and they are going to bundle together. So in motor neuron, there are 3 bundles of the neuron, and those are called column of Turney. That's one bundle. Another is called lateral motor column, and then there is third one that is called medial motor column. Now, when they are in one column, when they are together, they have the same target. So even if you remove this neuron, so you say, okay, let's remove this neuron from here and put that neuron here. So you remove it from here, put it here. It will, so where a target will be, it will still go to the target that was specified by this column. So even if you move from here, so you say, for example, Mason is sitting there, Mason, you need to go to St. Louis, you need to go to Kansas City. Even after I take you from there, put you there, you will still go to St. Louis, right? So once you're in the column, you have a specificity for the target, that where you want to go. So now we want to talk about how they go, what they decide, how they go, okay? So we know that these columns are formed because they are having specific neural cadherin expressed on those, and they form bundle with the similar type of neural cadherin, and then their target is specified once they are in a group of neurons. Now how they go, let's talk about that. Okay, now think about so this is your developing limb bud. So limb bud is also developing as the neurons are developing, right? And then right here is this is your spinal cord that is also developing. So developing limb bud, so this is the dorsal side, this is the ventral side, and right here is the spinal cord. Again, this is also developing. But the motor neurons have formed by this stage. Okay. So I'm going to talk about two bundles of the neuron here. So right here are here another group of cells, and these are dermamyotome, and these are going to give you the body wall muscles. Body wall muscles. This group is going to be. Now, so what we have here is, let's say right here is your right here are the neurons of your lateral motor column. Okay, and right here are your neurons of medial motor column. Just talk about two columns and let's see. Now, this lateral motor column has actually two subdivisions. It can be divided into two areas, this area here and this area here. Okay. Now, this area, now we will have motor neurons here. These are all motor neurons here. These are all motor neurons here. All motor neurons here. All motor neurons. Everywhere you have the motor neuron. So this area is called, so since I said there are two areas right there, right? So this area is called medial subdivision, and this is called lateral subdivision. Now, this is the limb bud. We are showing three group of neurons there. One group of neuron is, one column is lateral motor column, subdivided in medial subdivision and the lateral subdivision, and then you have the medial motor column. Now, how these neurons decide where to go? Okay. Now, we said that the surrounding tissue where they are going, that should produce the trophic factor where they want to go. So this is producing trophic factor. This area is also producing trophic factor. But right here on this side of the limb bud, here there is semaphorin produced on this side. On this side is afferent produced there. Okay. Now, the target specificity how that is determined, how they know where to go. So right in the beginning, when these neurons are formed, either from this group or these two groups. These neurons, all of these neurons, they start expressing two transcription factors called islet 1 and islet 2. They are present in all of these neurons. Okay. So, if these neurons are expressing throughout islet 1 and islet 2. So let's see this group of neurons, medial subdivision of the neuron. These group of neurons are always having islet 1, islet 1, islet 2. These will also have islet 1 and 2. And these will also have islet 1 and 2. Right from the beginning they are expressing this. First islet 1, then islet 2. Okay? So this group of neuron, the islet 1 and the medial subdivision neuron, they also start expressing limb 1. And when they start expressing limb 1, what happens? From here, these neurons, they move here. Why these neurons, their axon move here? What can be the reason? Why they don't come here? What do you think? Afrin is a chemorepellent, right? Semaphorin is also a chemorepellent. So they choose semaphorin instead of afrin. What can be the reason?
Like receptor.
Yes, receptor. So these neurons, which are in medial subdivision, they go to the column which is secreting semaphorin, because these neurons express neuropilin receptor. Neuropilin is a receptor for wait a second. Yeah, they don't express neuropilin. They do not express, these are coming here, they express EPH receptor. There's EPH receptor. So they are afraid of afrin because they have EPH receptor, so they cannot go there. Whereas these neurons, which are coming from here, these neurons, they go to afrin, and they keep on expressing islet 1 and islet 2. So that means the neurons which are in this column, they are expressing neuropilin. So if they are expressing neuropilin, they will not go to the semaphorin. They want to go away from it. Now these neurons, what they do, they send their axon out, but they don't either go here nor there. What they do, they actually reach here. So these neurons here, the medial motor neuron, they were expressing islet 1, 2, islet 3. They also start expressing LHX3. And when they express that time, at that time, neither they express neuropilin nor they express afrin. But what they express, these dermomyotin here, they secrete FGF. FGF is a neurotrophic factor, and these neurons, they want to have this FGF, and they start moving towards the FGF receptors. So it means these neurons here, they express FGF receptors. So let me go back everything so that things are very clear. So we are saying that these motor neurons, when they are made, their target specification occurs while they are being made. And we are saying that target specification has two components. The first, in which group those neurons will be arranged. And we are saying that depend upon the motor neuron and the cadherin that is specified on that. So neuron bundle based on the cadherin data. Once they are in a specific bundle, they know where they want to go. And then we are saying that how that specificity is selected. So we gave example here of the two columns. We said the lateral motor column and medial motor column. These neurons are there. These neurons of this lateral motor column, they are specified to go to the developing body. And we are saying that all these neurons, all the column, they initially start expressing islet 1 and then islet 2. So all these neurons will have islet 1 and islet 2. Then we are saying that this limb bud, it has semaphorin on the dorsal side, and on the ventral side it expresses afferent. And we said these are the signals saying come and go, right? These are the stop signals. These both are chemoreferent. So what happens in the medial subdivision, this medial subdivision also start expressing limb 1 in addition to islet 1 and islet 2. And when that happens, these neurons go straight to the semaphorin column. Now they don't go to afferent column. They don't go to afferent column because they can sense that there is afferent. And how they can sense? They should have the receptor for the afferent. So they start expressing EPH receptors. So they do not go to this side. Instead of that, they go to this column. Making sense? Now, the cells which are here in the lateral subdivision, they only express islet 1 and islet 2. What they do, they go to the afferent column. They go into afferent column because they cannot sense afferent, because they don't have the receptor EPH. But they are not going to semaphorin. They are not going to semaphorin because they start expressing the receptor neuropilin on that, and neuropilin can sense the semaphorin. So they avoid that area. And then the medial motor column neuron. These neurons are also expressing islet 1 and 2, but they also start expressing LHX3. And when they express that, these neurons neither express neuropilin nor EPH, but they start expressing the FGF receptor. And right here, the dermamyotome, which is going to give you the body wall muscle, that actually make MGF. So they can sense it and they basically move toward that. So you can see here, motor neurons are there from the same area there, of course from different column, but their target is different. But their target was specified not because of this, but because of the limb family protein and Hox family protein which are being made there, right? You have this islet 1, 2, what kind of, either you have LHX or limb, or you do not have any limb or LHX there. And then, once that target is specified, they are following a special area where they need to go. And that area depends upon the chemoattractant. FGF is a trophic factor attracting them. Come and take it, like money is there, right? So, they do not have any receptor for semaphorin or afferent, but they have receptor for FGF. So they basically go toward these. Now these cells which are here, they are coming to the semaphorin area. That means they don't have receptor for semaphorin, but they have receptor for afferent. Now these cells which are coming here to the afferent area, that means they have receptor for semaphorin, but not for afferent. That is making sense? So now, if I take a neuron from here and put it here, where it will go?
To the place.
It will still go here, right? Because it has FGF receptor there. If I take this neuron, put it here, it will still come here because it has receptor for semaphore. Making sense? Okay, don't put this for a minute. That will see what we have.
Oh, bud. Is this the body around it? This must be the body around it.
Yeah.
So when is the pilot one?
I think like in here, this one, like
They're
get it
not expressing, no, locally, they're not expressing, the EPH, right? So they're neither expressing this, but they start expressing the FGF receptor. FGF receptor, so they basically go to the. Making sense?
See, I don't have that issue.
Any other
The same
questions?
page, this way.
Mine doesn't say.
Mm-hmm. What is the conclusion?
No, I think we're good.
Good, okay. Any confusion here? We're good? Confusion, this one?
Is it the same way for Limb 1 as it was with LHX?
Yeah, so when you have limb one, where is the limb one? Limb one is expressed in days, right? So at that time, it is starting to express, it doesn't express the FGF receptor. At that time, what is expressing? It is expressing the EPH receptors. So depending on which transcription you actually have, that particular type of receptor you start expressing, and you start responding to those signals.
That makes sense.
For example, think about if you had a car, and that car can sense the car in front of you. That can sense only if you have a camera on the car. Then you have a car which can sense the lane change, should have camera in that area. Same if you have a receptor, particular type of receptor, then you can see that particular type of signal in there, right? That's how it goes.
Thank you.
Any question here? Okay, good. So we don't have time. 10:02, okay. So we have one more topic left we'll talk about. So far, think about, here is an interesting thing, just to give you overview. So some neuron from here, they will come, they will cross here. Some neuron will come, they will cross here. Right? So think about, that's what people usually say, your left hemisphere control right, right hemisphere control left, right? So how that crossing occurs, if you're saying all those things are there, right? And here I want to give you just an idea. In this lane, in the center, they're all inhibitory things there. Nothing should cross this from this, but somehow it crosses. And how that happens, we're going to talk in the next class. Okay, so we're done, and make sure that we are having the good drive for tomorrow, so that we can basically.
So we're meeting at 2:30?
2:30 in the lab. 2:30 in the lab.